starting with the name of Almighty Allah, the gracious and most merciful. Today now we will starting the lecture number 6. Topic is chromatography and chapter number 1 biology. Lecture number 6. Topic is chromatography. Chroma mean color. Graphy mean to write. Chroma mean is color and graphy mean is to write. Definition is the technique which is used to separate the dissolved solute from a solution called chromatography. Solution contain two or more than two components. Two or more than two components. Chromatography was used to separate color component. Color component. Two types of chromatography. Chromatography. First one is paper chromatography. The second one is column. It's a mobile phase. It's a silica. Mobile phase H2O. Chalk powder. The chalk powder is, it's a beaker, its depth in silica and pulpil dot is mobile phase. Mobile phase mean H2O water. It's a mobile phase. It's a silica. Mobile phase H2O. Chalk powder. The chalk powder is, it's a beaker, its depth in silica and pulpil dot is mobile phase. Mobile phase mean H2O water. After this, the paper chromatography. Column chromatography. Vapor chromatography mobile phase H2O and the column chromatography chalk powder of silica. It's a paper chromatography. Now discuss the column chromatography. Column chromatography. Column chromatography. First one is and second sample paper. Third one is large. Now pulpel the chalk powder. Pulpel the area